Culturing fibroblast in spheroid recapitulate tumor tissue much better than the commonly used 2D system on stiff plastic. It allows to study fibroblast differentiation in a tumor-like environment. In this method, the rigidity of cell culture plasticware does not artifactually influence fibroblast differentiation. Instead, the influence of cell matrix interaction on tumor-induced differentiation of fibroblast can be analyzed in vitro.
To begin this procedure, obtain the cell culture media, which is MEM supplemented with 1% heat-inactivated FBS and 1% penicillin streptomycin. Also obtain a 6 mg per milliliter methylcellulose solution. Mix 1 part of the methylcellulose solution with 3 parts of the cell culture media to prepare the spheroid formation solution. Resuspend freshly thawed immortalized normal fibroblasts or other cell types as listed in the text protocol in the spheroid formation solution. If cytokines or integrin inhibitors are included in the study, add these compounds to the cell suspension in order to embed them into the spheroids during their formation. Where appropriate, add inhibitor compounds together with 10 nanograms per milliliter of TGF-beta1 to trigger differentiation in immortalized normal fibroblasts. Distribute 100 microliters of the spheroid formation solution into each well of a round bottom 96 multi-well plate. Pipette the solution in each well up and down several times to mix. Then, incubate the plate in the cell culture incubator at 37 degrees Celsius with 5% carbon dioxide for 24 hours to allow one spheroid to be formed in each well.